All right. Hi, everyone. Welcome to day 2. I am just going to introduce our next speaker, who is Daniel Chafamo. Daniel, am I saying your last name right?
Yeah, that's right.
Okay, Chafamo. Daniel, you are a graduate student, I believe, in Alex Shalik's lab at MIT, and he is going to be giving our next talk. So thanks, Daniel, and over to you.
Thanks, Christine. Hi, everyone. Can everyone see my screen?
We can, you just need to put it in presenter mode now. There you go. Yep, perfect.
Awesome. Thanks, Christine, again, and thanks everyone for attending. I am going to be talking to you today about pre-processing and quality control of single-cell RNA-seq data. So we're going to go over the steps to go from raw sequencing files to count matrices first. I'm then going to give you a demo of Terra, which is a platform that we use to do most of our pre-processing. And then we're going to go over actual quality control that we do. And then finally, we're going to discuss normalization as one of the steps for quality control. So I hope yesterday you got a good idea of a high-level overview of how we go from cell samples that we collect to preparing the libraries and then sequencing them. So that's like the wet lab part of it, and that's how this whole process starts. The first silicon data object that we get from this whole process is the BCL, which is the output of the sequencing machines. And so I'm going to give you a high-level overview of what we do to go from these BCLs, which are base call files, raw like ATCG files containing that raw data, and we want to go to cell by gene matrix. Let me quickly flash that for you. So this table that you see here is what we want to work with. So this is the input for all of our downstream processing. So we want to get here for every cell, we have count for every gene. So this is the hallmark table that we want to get to, but we start from these raw base call files. So like the intermediate process is what I'm going to discuss first. So the first step is to convert those BCLs to FASTQs. FASTQs are a file format that is universally transferable. So it's like a sequence, a file format for carrying sequence information. So this is what a simple FASTQ file looks like. First, it has a sequence identifier, which has information about the cell, about the unique molecular identifier, and other information about the sequencing that is done. And then it has an actual sequence of bases. So here, this sequence has A, G, G, A, and so on. So these are the actual bases that were called. Then there's this separator sign, and then quality information about each base. So this base A has quality at, so we will be able to translate it using ASCII code, but like it's not really super relevant here. It's just as long as, you know, this is a quality measure. So this is, we can have like these 4 lines repeated millions of times to relay millions of sequences. So this is the FASTQ format that other tools can use and understand. So the first step is to convert those BCLs, which are outputs from the sequencing, to fast queues. And in order to do this, we can use tools like CellRanger MK-FastQ, BCL to FastQ, or BCL convert. When we go into the Terra demo, I'll show you CellRanger MK-FastQ, but you can use any of the other steps as well, the other tools as well. Okay, so we went over the FastQ. So once we have this FastQ, which is a set of these raw sequences, just like this, the next step is to align those sequences to the reference genome. So we have this sequence, for instance, right here that you can see as read, and we want to know which gene that sequence possibly came from. So this we use a reference genome that is compiled over time for different organisms. So for instance, for humans, we have sequenced and put together the entire genome, the human genome. So we have some idea, we have a reference genome that gives us an idea of what at each chromosome, at each location, what the base value is. And so we take that read and try to match it to a place in the reference genome where it aligns most fittingly. And based on that, we can assign this read to a region of the genome, and using that, know which gene that came from. Right, so that's called alignment, and there are different tools that we can use to align our sequencing data. The most common one I'd say is STAR. That's the one we use at least for CellRanger count, which is probably the most common tool for 10x sequencing data. So that's one common one, but there are a lot of different ones with different pros and cons. And if you are looking to use an aligner, there are different things you would take into account. One of them that was brought up last time is the amount of compute resources it takes and the amount of time it takes. Tools like Callisto and Sailfish take less time and less compute resource. So if you're working in a resource restricted environment, then you may consider using these two. Almost all of the resources are free to run, but running them using high performance compute clusters might be costly. So this is one consideration that you may have. Another consideration when using aligners is we want to use splice-aware aligners. STAR is one such aligner. Splice-aware just means when aligning reads to the genome, there are exons and introns, and a splice-aware read is able to map across those spliced out regions of the genome. That is very important, especially in animal genomes, which have widespread splicing. So that's all about alignment. So finally, once we have aligned our reads to the genome, we have an idea of where each which gene each read came from. We can now finally assemble our count matrix. So the way we do this is each sequence, as I showed you earlier, will have a cell barcode and a UMI, unique molecular identifier. So a unique molecular identifier, I think it was mentioned yesterday, will say that this sequence came from a specific molecule, and every sequence that you see as with UMI CCCT should be counted as just one molecule. So there are amplification steps in the sequencing process, and we don't want those to bias our counts. So we have this unique molecular identifier. the cell barcode you see here, for instance, GGTCCA, uniquely identifies a given cell. So we know that this sequence came from cell GGTCCA, and we'll have a bunch of these sequences which we have aligned to genes. So this may correspond to, say, gene 1, cell this. And so we go to our table and put that value for that gene in that cell column. So in this way, we produce this table, which is very important to understand properly. So for each cell, for each gene, this is the count that we have. We have 3 gene 1s in cell 1. That's how you'd read something, a table like this. Okay, so now we mostly do all of these steps. They're very compute intensive most of the time to convert our raw BCL files to FASTQ, to align them, and then to create the count tables. And so we use a platform called Terra, which is built on top of Google Cloud, also supports Azure, but basically what it does is it provides us a very easy to work with interface to run compute and memory intensive workflows in a repeatable and shareable manner. So if can quickly, yeah. So this is Terra. You can go to terra.bio and you should be able to log in and to sign up for an account with your Google account, I believe. And so the basic process of using Terra is mostly with workflows for our purposes. Workflows are basically a sequence of commands that are going to be executed based on a given input and will generate a given output. So for instance, this is our cell ranger workflow. We can provide it different inputs. So these inputs will tell the workflow where our raw base call files are, and then different parameters that control the processing workflow. And then once it's done, it will output the results in this output directory. So you can play around with the scripts. There are a lot of publicly available workflows. But basically, yeah, it's a very straightforward, very easily repeatable pipeline. So once you master how to run these pre-processing steps from BCL to count matrices, it will be as easy as just running, clicking this run analysis once you set up the workflow. So it's it makes the process very easy. So the output of, so one thing that we use often is cell ranger, like I mentioned, cell ranger, both mkfastq, which makes FASTQs, and count, which aligns to the reference genome and generates counts matrices. We use cell ranger for both of those steps, and the results that are generated look like this. So this is one output of cell ranger count, and you can see here this filtered feature BC matrix. This is the count matrix that I told you about, and oftentimes this is what you would start your downstream processing from. So you take this H5 file or the raw H5 file, and you can read it into Seurat, ScamPy, whatever your downstream tool is. You'll start from this. And another really cool feature of cell ranger is that it generates this web summary.html file for us, which tells us, which gives us a high-level overview of the results of the sequencing. So for instance, here we can see we recovered 2,900 cells with median 2,100 genes per cell and this many reads per cell. So these are all important information. The first thing we look at after we do these pre-processing steps is to look through these details. So for the question from Modinat, do I need to personally input codes in Cell Ranger or are they all default? So not code, so you don't need to like write any in any programming language or anything, but you do need to provide some mandatory inputs. Here it's just the location of your BCLs, the reference genome that you used. Is it human? Is it mouse? And there are other details of the sequencing, like the index and the chemistry of the sequencing that you used. So all of that information you have to provide, but the code is mostly pre-written, like it's already, it's like a function. You can think of it as a function. You just need to call that function. So you just need to give it the input and run it. That's all. You can, in the future, build your own workflows, and it's a very good tool, Terra, to build your own workflows as well. But for Cell Ranger, just provide inputs like and then run. Okay, so back to the summary. So yeah, I was mentioning how important it is to look at these values. This plot of UMI counts by barcodes, that's called a barcode rank plot, is also a very important thing to look at when you're pre-processing. So it's showing the number of UMIs, the number of, you can think of it as the number of RNA molecules recovered. And then these are the cells ordered by the number of RNAs we found in them. So for cell 1, we had 100,000 unique molecular identifiers. And for cell 1 million, or barcode 1 million, it's not actually a cell at this point. We only had one RNA count. So we expect to have a plateau, so like a straight line, and then a quick decline, a quick drop off, and then another plateau and another quick drop off. So Cell Ranger itself has a cell calling mechanism built into the sequence pre-processing steps. So it pre-calls for us which barcodes are cells and which barcodes are background noise. So in the droplets, so in different technologies, for instance, in the droplet sequencing technologies, there could be empty droplets that will have a barcode, a cell barcode, but they don't actually contain any cells. So that's what this background value is, but we want to work with cells only. So let me go back to the presentation. So yeah, a very cool thing with Terra is you can sign up with your Google credits and you'll have $300 of free credits to work on Terra. And usually the cost of running one, let's say, pre-processing step, it depends on the size of your data, but it's I'd estimate it to be in the less than $10 range for one sample processing. So this might take you, might give you a lot of mileage for getting started on Terra. So you can follow this link and read more about how to sign up and get access to the free credits. Okay, so this is what I was mentioning with the barcode rank plots. So you want to see something like this. This is a healthy processing sample. We have two plateaus, two knees, and the line drops up very quickly, like very precipitously. Others, so something like this, it's a sign that the the sequencing or library preparation had some issues, and the quality of the data that we recovered might be very poor. So for instance, on this one, you can see only 100 barcodes have any meaningful level of UMI, everything else has less than 100. So this is probably a failed sample. So the very first thing that you'd look in when processing is this barcode rank plot, and you make determination about the quality of your samples. This is just referring you to the Surat read 10x h5 function, which just directly uses that .h5 file that I showed you that's output from Sarranger. So it's so optimized at this point that all you need to do is plug in those inputs in the Terra workflow, run it, and at the end you'll get an H5 file, and it's on to Surat with this function. So it's very streamlined at this point. Okay, so now on to quality control. So this is the first step that we do on our actual count matrices. So once we get our actual count matrices, this should always be the first step. So the goal of quality control is to filter our data to only include true cells that are of high quality. So we often have in our data a lot of impurities and low quality cells. So we want to remove those. This often includes removing empty droplets. So these are the ones that I showed you earlier down here. Things that are not cells, things that are just empty might be called cells, and we want to remove those. We want to remove doublets. Well, we'll discuss more about those later. We want to remove dead or damaged cells, and we want to remove ambient RNA. We'll dig deeper into each one of these. And running quality control is challenging because it's hard to identify if a given cell has poor quality, or is it from a less complex cells. And by that, I mean, is it a cell that just has a low number of RNA and low number of unique genes, or is it actually a cell that has those low metrics because of sequencing issues, or just randomly happening to have low quality? So it's hard to differentiate these, so we will try to be careful and reason deeply about each of the filtering steps that we apply. So the first step in filtering is to use these three metrics to visualize our data and filter based on them. So we use number of reads or UMIs detected in a cell, number of genes detected in a cell, and the percentage of mitochondrial expression. Number of reads just means the total number of RNA that we found in a cell. So basically in this count matrix for cell 1, we sum up this entire column. So 3 + 2 + 1 + 25, and doing the same for all the genes, of course, we get a count for the total number of RNA that cell has. So if that value is too low, that kind of tells us that this cell, it might be an empty barcode that just happened to have some RNA that's floating around but not contained in a cell. We want to filter out any cell that has very few genes, it won't be very informative for us in any of the downstream processing analysis. So we just want to filter cells that have too few RNAs in total. And having too many, so like if this was too high, if the total number of RNAs was too high, we would also want to filter that out, because that's an indication that that cell might be a doublet, or like multiple cells caught up in the same droplet. So those are the two things, that the low threshold and the high threshold, that we will set for number of reads detected in a cell. Number of genes detected in a cell is also similar, but here, instead of summing up all the counts, we sum up, we just count the genes that have at least one value in them. So here in cell 2, gene M has 0 counts, so we don't count gene M, but gene 1, 2, and 3 have positive counts, so we say this cell has 3 unique genes. So that's what number of genes is referring to. And that also functions similarly to number of reads. Very low number of genes detected kind of indicates that it's empty and low quality. Too high is probably a doublet. So we filter on those criteria. And then percentage of mitochondrial expression, it's the fraction of counts from mitochondrial genes as a fraction of like the whole number of RNAs in that cell. And what this tells us is if the percentage of mitochondrial cell genes in that cell is too high, it indicates that that cell was damaged or dying, because the mitochondrial genes are kept in inside the mitochondria and are not lost easily, whereas the cytoplasmic RNA will escape easily if the cell is damaged. So the higher the mitochondrial expression, it indicates to us that that cell might have been damaged. So we also filter out any cell with too high of a mitochondrial percent mitochondrial RNA expression. So it's also sometimes helpful to visualize these metrics in conjunction. So here we visualize both percentage of mitochondria and end count RNA, which is like the number of UMI, the total RNA count. And feature RNA is the number of genes. So these are the names that are used in Seurat. So these are what you'll be looking at in your lab. So that's why I use them here. But yeah, so number of UMIs and number of genes. And we can see that some cells have very high in both number of UMI and number of genes. Same with the percentage of mitochondrial and number of end count RNA, which is number of UMI. So visualizing them together, we can identify these outliers and filter them out from our count matrices. Okay, another helpful thing to do in doing our QC is we should often visualize the QC per group. So here we have different samples from which this batch of cells came from, and we could visualize the number of genes per cell and percentage of mitochondria all together, but it's also helpful to see per sample what it looks like. So here we can see, for instance, the percentage mitochondria of control participant 15 is very high. And if we filter out using the same threshold for all of them, we might be removing too many of these cells. So it's often a good idea before you actually do the filtering step to visualize per sample and often per cell type what these metrics look like. Okay. The other key thing to keep in mind is the actual threshold. So you might be wondering what what value should I actually put the cutoff on? Is it 200? Is it 400? So that value often depends on a lot of variables. So it's hard to say a very strict cutoff. For instance, different platforms have different expectations. So SmartSeq 2, for instance, which is one of the sequencing technologies, often yields more genes per cell than 10x. So if you're working with 10x, the filter that you might set for your genes per cell might be different from if you're working with SmartSeq. Similarly, different cell types have different expectations. For instance, immune cells normally have lower number of fewer genes detected per cell than non-immune cells. You can see that T cells and B cells have a noticeably lower number of EMI per cell than these other cell types. So again, you may want to set your threshold. So like a T cell with, let's say 500-ish number of EMI might actually be a true cell, and we don't want to filter that out. Whereas at 500 number of EMI per cell, the endothelial might actually be an empty droplet. So different cell types might set different expectations. Again, tumor cells especially, you would want to avoid filtering tumor cells with too high number of genes or too high a number of UMIs, because often tumor cells can become very large, and these are probably not doublets, even though they're outliers. They're most likely just because the tumor cells are large. And a final example for percentage mitochondria, again, cardiac cells which have a higher number of mitochondria to begin with will definitely have a higher number of mitochondrial genes per cell than other cells. So again, if you have cardiomyocytes, for instance, here, and you're visualizing the percentage mitochondria for different cell types, and you try to set a uniform 5% cutoff, then you'd be filtering out almost your entire population of cardiomyocytes, which would not be right. So again, visualize the, going through the tips. The key to avoiding something like this is always visualize your QC metrics in a per group basis, per cell type, per sample, per whatever differentiating property that you have for your cells. Quickly visualize them before setting your cutoff, before setting your threshold. Okay. And yeah, it may be often necessary to revisit these decisions multiple times. So at first, when you do your QCing, for instance, you often do not have cell type information. So you'd start off with a more permissive. So for instance, we can start with like, it seems like here they started with like a 30 cutoff, maybe 30% cutoff for the mitochondrial genes. They went ahead and assigned the cell types and then visualized them. And then maybe we can reconsider for these cell types lower or more stringent cutoff. So it's a cyclic process where you go to the downstream analysis, come back to QC, and rinse and repeat. Okay. So the other QC process that we do is to identify and remove doublets. So a doublet is 2 or more cells that are captured together. And so the information that we get in our count matrix, even though it appears with a single cell barcode, it actually comes from multiple cells. So here in this doublet figure, you can see that 2 cells have been captured with this droplet, and when it's sequenced, they'll get the same cell barcode, but we're getting the genes from two cells in this case. So this will bias our analysis. So we want to identify these doublets and remove them from our count matrix. So this is just to give you a sense of the doublet rate. Often when we have very few cells, so this experiment just pulls together mouse cells and human cells, sequences them, and the plot we see here is the number of UMI counts for mouse and the number of UMI counts for humans. So if the values are on either the x-axis or the y-axis, that means they're singlets. But if they're in this in-between space, that means they have some mouse genes and some human genes, which indicates to us that doublets have occurred. And this is this cool experiment they did to identify the rate of doublet or multiplet formation. And you can see that at about 1,000 cells, the multiplet rate is 0.5%. But when you have, let's say, 10,000, then the multiplet rate is like 5.4%. So it's very crucial to identify these and remove them if you have a large number of cells. So the first thing that we notice about doublets is that they have high total UMI counts, which makes sense because we're pulling two or more cells together, the genes from two or more cells together. So we obviously would expect to have a high number of counts. And that isn't, we already mentioned how we filter by a high threshold to remove potential doublets. This doesn't necessarily mean that all cells that have high UMI are doublets though. So if you see this figure on the right, we can see that there are, so the singlets are gray, the doublets are red, and the x-axis is the total counts. And you can see that there are, there are singlets that have total counts higher than some doublets. And so like setting a very a hard threshold might not be the solution because you'll potentially lose singlets as well. But it is generally a good indication that high total counts, if you see a cluster that looks like a doublet and it also has high UMI, that's a confirmatory evidence for doublets. There are also tools like Scrublet and Doublet Finder that use simulations to assign doublet scores to cells. So these methods simulate doublets, so they pick up singlets from your single cell data, mix them to create simulated doublets, and the cells that look most like the simulated doublets will be assigned a doublet score. So we don't need to go into detail about that method, but basically we can use these tools like Scrublet, Doublet Finder in conjunction with the number of UMIs, and also using marker gene overlap, which just means if a cluster has genes from both, let's say, T cells and B cells together, that's also an indication of doublets. So we can use these various indicators to identify doublets and remove them. Okay, and another QC step that we do is to detect empty droplets and correct for ambient RNA. So what we mean by empty droplets are droplets that do not have any cells in them. And ambient RNA just means RNA that is, so in the surrounding the cells, there's RNA that is not enclosed in any cell. So this can be coming from dead or damaged cells that just leaked out. And it gets captured, as you see in the figure here, it gets captured alongside actual cells. So when we do the sequencing, the RNA for this cell will have these random floating RNAs as well. So we want to, if we can, identify these and remove them, and that's called ambient RNA correction. And we have this tool called Cellbender, which really helps in detecting empty droplets and correcting for ambient RNA. It learns the background noise and artificial counts from non-empty droplets, and it removes the ambient RNA that it modeled from cells that are going to be kept in our analysis. So this is an example of the transformation we get from using Cellbender. So it's especially very helpful in making our clustering very clear and removing ambience from important marker genes. So here we can see LYZ gene is a monocyte marker, but we can see there's ambient expression due to so B cells often don't really have a significant amount of this gene, but just because we have ambient RNA, we can see that it has some counts. But after correcting by Cellbender, we can see that the number of Liz in all these other cell types goes down, and the true monocytes will have higher LYZ gene counts. Okay, so that's all about QC, and the next step of our processing is normalization of gene expression data. So normalization just means adjusting the raw data to account for unwanted factors that prevent direct comparison of expression measures. So when we get our counts, our count matrix, we cannot always directly compare one cell to another cell. So it could be that the sequencing depth, so the number of the total number of reads that are sequenced for a cell are just higher than another cell. So there's nothing that keeps the total counts from being different. So they could be different due to natural reasons, biological reasons, but it could also be due to technical reasons. So we want to control for that, and that's what normalization allows us to do. So there are two steps often when we talk about normalization. The first one is scaling, and the second is transformation. So scaling just means dividing our counts by the cell's total count. So for a given cell, if we have, so let me go through the count figure. Yeah, so here for this cell, for each gene, we have this counts. So we sum up all of these. So 3 plus 2 plus 1 plus 25, and we divide each of the counts that we have. So for instance, gene 1 is 3. 3 we divide by the total, which is 31. So 3 over 31 is our now normalized count. So we do this for each of the genes. We divide them by the total number of RNA that we have. In that way, if a given cell was sequenced very deeply, it won't affect us because we have already divided out the total number of RNAs. And then transformation just means applying, and often it just means applying log 10 to our data so that the distribution resembles, aligns with the expectation of some downstream analyses. So, so this is the effect of our log transformation, for instance. this is the total expression before normalization, which, as you can see, is very skewed. And oftentimes it's very zero skewed as well. And then after normalization, we can have something that resembles more normal data. So why normalize? One of the things here, as I mentioned, is that it corrects the distribution for us so that it's more in line with downstream analysis tools. And the other one is, especially for the log transformation, we want to think of our count values in terms of fold changes. So for instance, if you see the difference between cell type A and cell type B, so when we go from 1 to 2, the difference is 1. When we go from 100 to 200, the difference is 100. This reflects changes in raw data, but for our perspective, from our perspective, we see that it just doubled. So gene 1 doubled from cell type A to cell type B, gene 2 doubled from cell type A to cell type B. So this difference in scale can be hard, can make interpretation very hard. So by log transforming our data, we can see that a shift from 1 to 2 is a twofold change. A shift from 100 to 200 is also a twofold change. So it equalizes these extreme scales for us. Okay, the most common normalizations that is protocol that is done is called count depth scaling, also known as CPM normalization. As I mentioned before, all we do is normalize by the total gene expression. So we divide by that total amount. And then we usually scale that to sum to 10,000. So this is just to make it so that we don't have low fractions. So like we don't want our counts to be 0.001 or something. We want, it's just to make it better for visual reading. And then finally, we log transform. CPM is this value, counts per million or counts per 10,000 or whatever that we found. We do +1 because to avoid getting log of 0. And yeah, we just log transform this normalized value. So that's the most commonly done log transform normalization. So there have been a lot of questions about the appropriateness of this kind of CPM and then log CPM normalization. As you can see here, especially when the values are 0 inflated, it distorts the data to look bimodal here. So this data that we started with ends up looking like this. We like the fact that it's looking more normally distributed, but there's this huge difference gap created between 0 values and non-zero values, which might affect our analysis. So there are more advanced tools like SC transform that try to model the data better and get a more less distorting kind of normalization. So if you want to look more deeply into it, look up SC transform and similar methods. Okay, that is all from me. Let me know if you have any questions. We'll go more deeply into it in the lab as well. So if you have questions, then I can address them then too.
Thank you, Daniel. So if there are any questions for Daniel, please feel free to either write them in the chat, or you're also welcome to take yourself off mute and ask Daniel directly. So we'll give it a minute or so for questions to come in. Okay, if there are no questions, we do have a break scheduled. We're running a little ahead of time. So why don't we plan, is Sarah in the room? Is
Yes,
Sarah
I'm
here?
here.
Okay,
Yeah.
perfect. Sarah, are you okay to start 15 minutes early, so at 8:15?
Yeah, sure. That works.
Okay. Great, so let's go ahead and take, it will be a roughly 20-minute break, and we will reconvene here at 8:15 a.m. Eastern Time, which is 1:15 p.m. GMT. So see you back here in 20 minutes. Hi, everyone. Hope you all had a good break. The next we have here is Sarah Ingabire from the Shalick Lab. She is a research associate. And I just want to make a correction about Daniel. Daniel, our previous speaker, is a data analyst and software engineer in the Klarman Cell Observatory, which is located at the Broad Institute of MIT and Harvard. So now we will pass the baton over to Sarah. So Sarah, over to you.
Thank you, Kristen. Let me go ahead and share my screen.
Great, and I'm just confirming for you that I can see your R studio, it looks like.
Okay, great. Okay, so hi everyone, my name is Sarah. So today we'll be practicing what we've learned yesterday. So for I think what will be helpful will be like to just do it together so that when you go to the step of actually doing the QC lab, you actually have like some basic concepts and you know how to use R. So I think you can go ahead and open this website, the one that Vincent showed you yesterday. And when you open it, you can just double click on this introduction to R, or you can go to this file here and then do open file and then click on this file right here, the introduction to R. So please let me know if you have any question. Feel free to stop me at any point. Okay. So the first thing that we're gonna learn is how to create an object. So what I told you yesterday, we use the console to test any code that you want to test. So we can go ahead and test this. So we want to know like what is 4 plus 6. And then in order to run your command, you need to hit command plus enter, or you can click Windows plus R for Windows users. So when you click on it, you'll see that the results is going to be printed right here. So 4 plus 6 equals to 10. But what you're going to notice is that nothing will be saved in the environment. So your results won't be saved anywhere. And then to save your results, you basically need to assign the variable to your value. So you basically need to name it. Okay, so in this case, I named mine X. So I want it to be called X. And then we use assignment operators. So these can either be equal sign, or it can be this arrow right here. So I like to use this better. So I can go ahead and assign it. So I want my value to be named X, and then I retype what I typed earlier. So, and then I run my command. And then you'll see here that X has been saved. It has one element. and it has a value of 10 right here. So another thing to keep in mind is that R is case sensitive. So like I told you yesterday, so lower cases and capital cases are gonna be saved different as two different objects. So if I used X right here, but like instead of a lowercase, I'm gonna do a capital case right here, X, and then I redo the same thing. And then I run it. You'll notice that X has been saved as a different object, although they have the same value right here. Okay, so those are the things that you need to keep in mind when you're writing your codes. Another thing that we can do is to see how to list objects. So in this case, let's create more objects. So A equals to 3. It's going to be saved right here. B equals to 10. It's going to be saved right here. And then in order to view your objects, we use this function called list. So we simply do ls and then parenthesis. And then when you run it, it's going to show you a list of all the objects that you have so far, that you have saved so far. And then in order to remove your objects, we simply use this function rm. So you can do rm, and then you can specify a specific object that you want to remove. So let's say we want to remove A. So we can go ahead and type A in parentheses, and then when you hit Command Enter, you'll see that A has disappeared from the environment. You can also remove all objects. So you can reuse the function rm, which stands for remove, and then you list all objects that you want to remove. So in this case, I want to remove everything. So when I hit Command Enter, you'll see that everything has disappeared from the environment. Okay, so now let's go ahead and practice the data types that we learned yesterday. So R is a high-level programming language. It works with objects, and these objects are characterized by their names, their content or attributes, and they are constituted of basic types of elements, and those data types are known as integers, numerics, character, and logical. So integers are all the positive values, okay? So in this case, let's say x equals to 2. When you run it, it's gonna be saved right here. And then in order to check if x is an integer, you need to assign it as such, because so integers are also part of numerics. So remember, numerics are any decimal point, any positive, negative value, like, so numerics is all the numbers, okay? But integers are positive numbers. So if you ask, for example, like you do is dot integer, so you're asking R if this is an integer, it's gonna say that it's false. So in order to make it an integer, you need to specify as such, and we use the function as dot integer. And then you put an X, and then you'll see that, and then now we can redo the same function. So is dot integer X. Oh wait, 1 second. And then we can check the class. One second. So let's as dot integer Okay, so now let's go ahead and check the class of X. Okay, so in order to, what I was saying right here, so we, X is equal to 2 is gonna be, when you run it right here, you saw that at the beginning, it was a numeric type. So in order to make it an integer, you need to specify as such. And in order to do that, we use this function as.integer. And to do that, you basically have to rename X right here. So you need to assign the variable, okay? Tell R that, okay, I want to rename my X to be an integer instead of a numeric data type. And to do that, so I renamed it, I want it to be X, and then I use this function right here as.integer, and then I want my integer to be X. And then when you recheck the class again, this is going to be an integer. And then when you go ahead and look at your environment, you're going to see that X is an integer. It has 1 element and it has the value of 2. And remember yesterday we saw how, so each integer has this capital L letter. Like this simply means that indicates that it's an integer instead of a numeric data type. Okay. So now let's go ahead and see numeric data types. So numeric values contain any decimal points, positive values, negative values. It's like everything basically, like whole numbers. So here we have an example, n equals to 0.5. You run it. It's going to be saved right here. You can also name another one s equals to -1. And then it's going to be saved. One second. So here there is an arrow. Make sure to like use the right assignment operator. And then I want my s to be equal to -1. It's gonna be saved right here. And I want my m equals to 10. It's gonna be saved right here. And then you'll see what you'll notice that any like the negative values, the decimal point, the positive values, all of them have been saved as numeric. And you can also check the class by just typing class and then put in the name of your object. So we can maybe, let's check for the one that is negative. So we want to know the class of X. When you check it, you'll see that this is a numeric value. This is a numeric data type. You can also ask R, is this an integer? So let's suppose that, oh, we want to ask R that is S an integer, for example. So remember, integers are positive values. So this is going to return as false. Okay. So let's now talk about the characters. So the character values are anything that has to be in quotes, like in quotation marks, and feel free to use either double quotes or single quotes. Everything will turn out the same way. So we can, I named here my character that I want to create it as my character. So I assign my variable, and then I use the assignment operator, and then here my example is single cell is amazing. Okay, so when I run it, so you hit Command Enter, you'll see that in the environment, this has been saved as a character data type. It has 1 value, so this is gonna be count as 1 element. So yeah, and then it can be anything. Okay, so it can be numbers, it can be words, it can be like let's view another example here. So you can also, let me name another one, my character one, and then let's use single quotes, and let's use number like 12. So when you run it, you'll see that this counts as a character data type. It has 1 element, which this element is a number, like it's a number. So so like, yeah, like I said, it can be anything as long as it's in quotation mark. Okay. And another thing to keep in mind is that you cannot write you cannot put double quotes inside of double quotes. So like, let's say an example right here. So let's, for example, say my character 3, and so if you run like single cell is amazing, and then let's say you put another quotes right here, R is going to be confused and is going to think that you forgot to close some quotes. So when you run it, it's going to say it's an error. So if you really want to put double quotes instead of quotes right here, all you need to do is, let's copy the same example, and all you need to do is to follow it with a backslash like this. And then when you run it, R is going to treat this code as a special character, and this will be saved as a character data type with one element, and which is this one. Okay. And you can also check, is this a character data type? So just as we did for the numeric and integers, you can do the same thing for character data types. So you can simply do is.character. And then you can put in my character, which is what we created at the beginning. And then you can run it. You'll see that this returns as a true. So if you run both of these two others, you'll see that both of them are going to be turned as a character data type. Okay, now let's talk about the logical data type. So logical data type are the Boolean truth values. So it can either be true or false. Okay, so let's create another example called my logical. And then let's say this, I want my logical to be false. We run it, it's going to be saved right here, like as my logical. You'll notice that this is a logical data type. It has one element, which is false. So you can either write capital F, or you can either write F as false as in full words. And the same thing applies when you're writing true or true. So you can either write true in all the letters, or you can simply do T. And then when you run it, you'll notice that my logical right here that we just created here, like has one element with a true value here. Okay, so these are the examples that I wanted to show you in the case you're using these values. So let's say W equals to 1. And then let's say Y equals to 4, we run it, and let's name Z, and we simply want to know is W larger than Y. Okay, now let's print the value of Z. So you'll see that this is false. So you can redo the same thing. So now let's do the opposite way. Let's see if W is less than Y, and then you print the value of U. You'll see that this returns to true. Okay. So now let's talk about the factors. So factors are seen as categorical variables, and they are used when doing statistics or plots. So the same thing, the thing that you have to keep in mind with factors, so you have to like, so they are seen as categorical variables. So you need to put in a category. So in this case, I chose the sex category like I was showing you yesterday. So let's name it as my factor. And to create a factor, you need to use the function as.factor, as we've seen yesterday. And then you put in quotation mark, what do you want, like, what do you want your factor, like your category to be? So in this case, it's a sex category. So I'm just gonna put in male. I could have put in female as well. And when you run it, you'll see that my factor has been saved as a factor data type. It has one element with a factor with one level. So this male right here is going to be counted as one level. You'll see that when we're creating vectors, you'll see like what I mean by one level when we put a bunch of factors together. So another thing that we can do is we can manipulate objects. So basically, we just now we just learned about those data types, right? The integers, the numeric, characters, or logical. So we can put a bunch of numeric data type together. We can put a bunch of characters together. We can put a bunch of logical together. And when we're doing that, we're basically creating a vector. So remember that vectors are homogeneous. They can, meaning that they can only hold data of one type. Okay, so if you're planning to like put different data types together in one vector, certain rules will have to be applied to it. Okay, so let's say we want to create numeric vectors. So when we're creating numeric vectors, means that we're putting a bunch of numeric values together. Okay, so and to create a vector, we use the combine function C. So C is to concatenate, meaning that we're putting things together. So let's name our numeric vector as numeric vector, like this. So this is the name that I gave it, and let's create a vector of numeric values. And to do that, we use this function C. C stands for concatenate, and let's give it random values. So I chose 2, 6, 3, 4, 5, and then you run it. You'll see that numeric vector has been saved as a numeric, and the thing that tells you that it's a vector, you're gonna see this square bracket right here. Okay, and it has 5 elements, which are 2, 6, 3, 4, and 5. Okay, so now let's create a logical vector. So let's put a bunch of logical values together. So logical, so I'm named it a logical vector. And then to create a vector, you basically use the function C, and then you open parentheses, and then you can just put random logical values in it. So this is just an example. And then when you run it, you'll notice the same thing. So logical vectors, you'll see that it's a logical vector. It has 5 elements. It also gives you a list of these 5 elements, which are false, false, true, false, true. Okay. So the same thing applies for characters. So remember, characters, I told you that you need to code them to make it a character. It can be anything. It can be letters, it can be words, it can be a phrase, it can be anything that you can that you want to put in as a character. And let's look at the same thing. So here I named it as character vector. And then I use the function C to create a vector. So to tell R, oh, I want to create a vector. So I want to put a bunch of character values together. And here I gave an example of X and Y and Z. 27, 10, and I put in a random example. So let's say this is a phrase, okay? And when you run it, you'll see that, so I told you that each thing that you code is gonna be counted as one element. So in this case, we have 1, 2, 3, 4, 5, 6. You'll notice that here, this is saved as a character vector, and the thing that tells us that it's a vector is those square brackets right here. And then it has 6 elements, and it also gives you a list of those 6 elements. Okay, so we can also put a bunch of factors together. So remember that factors are seen as categorical variable. Remember that when we were viewing this example, so I chose my category to be like a sex category. So in this case, I chose a different example. So this is like, in this case, the category are alphabets. So let's create a factor vector of alphabet, okay? Let's first name it. So if you want to save your results, you need to name it always, and then To create a factor, remember we use this function as dot factor. And then if you want to create a vector, so if you want to put a bunch of factors together, you need to use the function C, which stands for concatenate, so means that you're about to create a vector, and then you open parentheses and you can start creating the factors that you want to create. So in this case, I chose alphabet. So let's go ahead and type in. And then we run it. So when we run it, you'll notice that this is the factor vector that we just created. It has 5 elements, which are A, B, C, D, E. So each letter is going to count as one level. So let's see a different example to see what I'm talking about. So let's, for example, run this factor vector 1. So when we run it, when we hit Command Enter, you'll see that this factor vector 1 has been saved right here. It's a factor data type. It's a factor vector data type, and it has 3 levels. So it has 5 elements still, but it has 3 levels because B is repeated 3 times. So it's gonna count level as A is 1 level, B is 1 level, C is 1 level. So this is gonna be 3 levels, but these 3 levels contain 5 elements, which are 1, 2, 3, 4, 5. Okay, so does anyone has any questions so far? Okay, so another thing to keep in mind is that when you're putting different data types together, because remember, vectors are homogeneous. So if you decide to put, for example, logical and numeric data types together, this is going to be converted to numerical data, numerical vector. Okay, let's, for example, see an example. So in this case, I named it so that it's easier to understand. So I'm about to combine logical and numerical values together in a single vector. So remember, to create a vector, we use the function C, and I'm putting in a numeric value and another value, logical value, which is false. So when you run it, you'll see that this logic num vector that we just created right here has been saved as a numeric vector. And the false value that we created here is going to be converted to 0. So if you choose to put true here, like true is going to be converted to 1 and false gets converted to 0. Okay. And then let's say we want to combine logical values, numerical values, and character values together. So let's name it like this, and give it an assignment operator. So let's create a vector by using the function C. So let's combine numeric values, in this case, true, 2, and then true. And then let's put in a character value. So characters are coded. So let's put in the X value. So when you run it, you'll see that we go and look at this thing that we just created. So this is the logic num character. So this has been converted to a character vector. It has 3 elements, which are 2, T, and X. And when you look here, this 2, which was supposed to be a numeric value, has been converted to a character. So it's coded now. And this true value right here, which is supposed to be a logical value, is coded as well. So these 3 are going to be converted to character data type. Okay, so these are the few things that you need to keep in mind. Since vectors are homogeneous, you need to keep in mind that if you're putting the different data types together in one vector, certain rules are going to be applied, which are these following ones that we just saw. Okay, so now let's talk about lists. So remember yesterday we viewed how list can be anything. So list can be a vector. You can put in numeric values in it. You can put in character. You can put in another list. You can put in a data frame. So you can put in anything that you want in a list. So let's, for example, list everything that we just the different type of vector that we just seen. So, and to create a list, we use the function list like this, and then you open the parenthesis. So we're gonna use the numeric vector, which we've just created right here. So remember, all the results have been saved in our environment. So if we simply put in the numeric vector, which is how we named it, we do tab, and then we put in the logical vector, this one. So all of these have been saved in our environment. So R will know what we're talking about because we've already created them. So we want to list the numeric vectors, logical vectors, and character vector that we've just created. So you hit Command Enter. You'll see that list right here has been created with a list data type, and then it has a list of 3 elements. So when you click on this magnifying glass right here, you're gonna see a list of these 3 elements. Okay, so it's gonna show you the numeric vector, which is sometimes called as double right here, and you're gonna see a list of these numeric vectors. So 2, 6, 3, 4, 5, which we've created right here. So remember, when we created this numeric vector, you'll see the logical vector that we've created and the character vector that we've created. Okay. So you can also put in a bunch of things in at the same time. So on top of this that we've created, we can add in a number. We can add in a list inside of another list, and we can also add the function right here. So I created another list, which I named my list 1. And when I run it, and then when we click on this magnifying glass right here, so you'll see that there is a numeric vector, the logical, the character, the number that we've added, which is numeric as well. You'll see that there is a list inside of this list that we've created. So if you click on this button right here, it shows you what this list that you've added inside of a list contains. So it has this list contains the same thing that we've added right here. So it has a numeric vector, it has logical vector, it has character vector, and it also shows you the function that we've added inside of the list. So yeah. So list, you can basically, one thing that you need to keep in mind is that list is non-homogeneous, and you can add anything that you want in the list. Okay, so now let's talk about the data frame. So data frame is basically, it's a list of vector, but they have to be of equal length, okay? So remember that here, for example, we've already created these numeric vector, logical vector, everything. So in order to create a data frame, you need to put vectors, but these vectors have to be equal length. So, and to create a data frame, we use this function right here, which is data frame, okay, data.frame. And I named my data frame as my data frame. So when you run it, you go ahead and in your environment, you'll see that my data frame has been saved as a data frame type. And it has, when you click on this table right here, it's gonna take you to another tab. And you'll see that numeric vector and logical vector. So the reason why this worked is because it has the same amount of elements. So each vector here that we've created has had 5 elements into each one of them. So if, for example, numeric vector and logical vector didn't have the same equal amount of elements, this wouldn't have worked. So let's, for example, see this example right here. So remember, so the numeric vector has 5 elements, logical vector has 5 elements. We can go ahead and check the character vector. So character vector right here has 6 elements. So it had, we can go ahead and check in the environment. So remember the character vector, it has 6 elements, which are X, Y, Z. So if we try it, it's gonna say that it's an arrow because these do not have equal amount of length. Okay, so another thing that you can do is you can name them. So remember, we've added numeric vector and logical vector, so we can assign them. I can be like, oh, I want my X to be equal to numeric vector, and I want my Y to be equal to logical vector. So when you run it, and then when you go to my data frame 2, and then you click right here, you'll see that X, which is the numeric vector, has been converted like to X, and you'll see that the logical vectors are now named as Y. Okay, so now let's talk about the matrix. So matrix is a combination of 2-dimensional objects with the same data type, okay? So in matrix, you just need to put in the data type that you wanna work with, and then you specify the number of rows and the number of columns. So when we run this, for example, that we've just created, so we go to my matrix, you'll see that my matrix has been saved as a matrix data type. It has 6 elements because it's a character vector. Remember, the character vector that we've created earlier has 6 elements, which are these elements right here. So if we want to view the matrix that we've created, you can simply you can simply click on this table right here, and this will take you to this. And you'll see that, remember, we want 2 rows and 3 columns. So this is gonna be like 2 rows and 3 columns. Okay. So we can also use indexing in order to access the values of an object. Okay. So let's say, let's go to logical vector. So we want to see the values of logical vector. So let's run it. So these are the values of logical vector, but let's say we want to see the second element. So in order to do that, we use the indexing. So you can simply do logical vector, and then we use to create to basically view the elements. You need to use the square bracket. So that's how we're using indexing, and then you click Command Enter, and then you'll see that the second element right here is false. Okay, you can also be like, oh, I want to know the first 6 elements, okay? So of which are in my character vector. So to do that, you simply do character vector, and then you use the square brackets, and then you can simply do 1:6, and then you click Command Enter, and then it'll give you a list of these 6 first elements. Okay, you can also choose to eliminate one value. So you can also choose, like, for example, this one is saying, oh, I want to know all my values except for the first one. So in order to do that, you basically put a minus sign right here. So when we run this right here, so remember the logical vector contain these values right here. So when you run it, you'll see that this first value has disappeared. So those are the kind of cool things that you can do with R. Does anyone has any questions so far?
was wondering if this last, sorry, so I was wondering if this this last way of manipulating the vector would actually change the element. So for example, now that we removed the first one, is it just, did we just remove it for check them without it, or is it actually?
Oh, no. Oh, sorry. No. It's not gonna be removed. So like, for example, if we can do, so we didn't remove the objects. Okay, we just, yeah. So if you view it, so let's say we want to view it again. So false is gonna be here. So we didn't delete it. We just want to see all the values except for the first one.
Thank you very much.
You're welcome. Okay. So now let's see how to create a function. Okay, so to create a function, you basically use the function here, like for example, like so this is an example that I gave right here. So I want to name it as new function. And you can be like, I want my function to have value 1 and value 2. And I want the results to basically, I want it to be calculated as value 1 plus value 2. So when you do that, when you return the results and you give the value that you want to use, so in this case, 15 and 20. So when you run it, so first we need to run this function right here that we've just created. Okay, and when you run it with examples right here, 15 and 20, you'll see that it's basically gonna take 15 plus 20. And you can do the same thing, okay? Like these are the type of ways that you can create a function. So you basically have to be like a function, give it value 1, give it value 2. Like in this case, they choose 1 and B. And you also have to specify what kind of function do you want to use. So in this case is just addition, and this is just the sum. And then in this case, it's just dividing, the dividing function. So when you do command enter and then you redo the same value, you give it the value 1 and value 2, you'll see that it's going to divide 15 and over 20. Okay, so you can do the same thing. You can do the same thing, and you can also like interchange the values. So in this case, for example, you can be like, oh, I want my value 2 to be equal to 15 and my value 1 to be equal to 20. Okay, so when you run this and then you run the new function, okay, yeah, you'll see that it's gonna divide 15 over 20. Okay, so now let's talk about ways of installing packages. So remember, R comes with a built-in set of packages, and these packages can be run directly from your R console. And so packages are obtained from the cron website, like we've discussed yesterday, and there are a collection of functions, and they can also be created by anyone. So here are a way of creating a package. You simply have to do install. You simply have to use the function install dot packages. And then you put in quotation mark the name of the package that you want to install. So I'm not going to run this right here because I've already installed the package. So once you've installed it, so it's gonna be installed and everything, and then you'll need to load the package. And to load the package, you simply use the function library, and then you put in the name of the package that you want to load. So, and then you hit command enter. And then if you want to see a list of all installed packages, you can simply do library. You can simply do library and then parenthesis. So when you run this line right here as command enter, so you'll see a list of all the packages that are available within the R right here. Okay. So you can also obtain help. So get information about the package if you don't know what it means or like how to use it. So you can simply do library and then package. So right now, like here, we want to know the ggplot2 information. So this tab right here has opened, and then it gives you like basically a description of what is ggplot2. It gives you the version. It gives you like all the information that we want to you that you want to collect on how to use it. So these are the all like these are all the like the objects that you can run when you're using ggplot2. Okay. So let's now see how to get help. So you can either to get help, you can use the function help right here, and then you put in the name of the object. You put in the name of the function that you want to get help with. So when you run this right here, you'll see that it's going to give you all the information that you want. It gives you the description, it gives you how to use it, and it also gives you like what each argument, like what does it stand for, okay? Like for example, X is an OR object. So this is how you use X and everything. So you can also do, if you wanna, so these are the two ways you can get help using this help right here. Also feel free to just type in, so if you go ahead and type in right here, mean, the same thing will reappear right here. Or you can simply do a question mark and then put in the name of the function that you wanna get help with. Or you can also ask R to give you an example on how to use this. And then it will basically show you like, okay, if you want to use the mean function, this is how you can use it, okay? So in R, you can also read data in a file. So if you're not, like the first thing that you need to do when you're using your data is to set your working directory. And if you're not sure of where you are, like of what your working directory, like where you are, where you currently are, you can simply run this code right here, which is get working directory. And then when you hit Command Enter, it'll show you like, this is my working directory. So, and then after you know the working directory, you can just go ahead and copy this to here. And then set your working directory to be like this, okay? You can also import files such as Excel files. So these are imported using CSV file, or you can also import the TSV file as well. So these are used, these are imported using this function right here. So red.dlim, and then you indicate the path to your TSV file, and then you use this code right here. Okay, so now let's see an example on how to use the ggplot2 function library that we've just learned. So the first thing that we need to do is to load the library. So in this case, we're using the ggplot2, which is popular for data visualization. This is the description of ggplot2. So it's a plotting package. It allows us to graph most kinds of data in a very simple way. It was designed by Hadley Wickham, and it provides helpful commands to create complex plots from data in a data frame. So the first thing that we want to do is to initialize the package. So we load the package by simply using this function, the library, and then you put in the name of the package. So I think we've already loaded it. And then in order to create a data set, you can, like when you're starting to use R, you can simply like, one of the things that helped me is first of all, like go and see, like view a list of R data sets. So this is a list of R data sets that are available within R. Okay, so I chose the BOD, which is the biochemical oxygen demand. So And to load these data, so if you're not sure what is BOD, you can simply do BOD like this. And then it's gonna show you the R data sets within BOD. Okay, so we're gonna load the biochemical oxygen data set. And to do that, so we've simply just created it. And then once you've chosen your data set, another thing that you have to do is to define an aesthetic mapping. So in other words, you need to specify like how would you like your axis to be represented in the plot, okay? And you basically have to specify the respective variables from the data set. So you need to, so when you're creating ggplot, let's just type right here ggplot, ggplot2. So yeah, you'll see that here you need to define your mapping, which is the aesthetic. So what would you like your X to be? What would you like your Y to be? Okay. So, and at this point, no plot will be printed until you add the geom layers. So what are the geoms? So the geoms, which are like the geometrics, they're used to represent graphics of the data in the plot. And there exist so many different geoms, okay? But the most one that we like to use are these, the geom points, which represent the dot plots or the scatter plots. There are geom line, which represent the time series, trend lines, geom box plot for box plots. So there are different ways you can plot your plot using ggplots. And in order, if you want it to be plotted, you need to add these geom layers. So in my case, I added the geom point and I want my size to be 10. And you can also define the color that you want. So let's run this code. So I named it biochem oxygen demand. I use the ggplot. My data set is BOD. These are the X that I've created. So I want my X to be time and I want my Y to be demand. And I added two geom layers right here. So I want my geom point, the size to be 10. So when we run this, and then we draw the plot. So we want everything right here that we've just created plus geom point to create the plot. So you'll see that this plot has been created, right? So this is the X, which is the time, and this is the Y, which is which corresponds to the demand. You can also try to change the size to see how or what is the difference. So if you change the size, this is gonna be like smaller. So because it went from size 10 to 3, okay. And so these are the ways that you can create plots using ggplot2. And please feel free to play with more geom layers or use different R data sets right here. So this is a list of all the R data sets that are available within R. And then you can just practice with different like geometrics, like different geoms. Like you can use the line, you can use the box plots, depending on what you want to see. Okay. So does anyone has any question?
I was wondering, wait, let me turn on my camera. I was wondering, so from now on or till now, we were working in the browser, and I was wondering how I could save this for my.
Oh yeah, so that was the question that you had yesterday. Okay, so do you want to transfer it to your computer, to your local computer?
Yes, for example, to use it from desktop version of RStudio.
Yeah. So to do that, you basically have to, so remember, if you want to work with the COVID data sets that we're going to work with today, so you'll need to also export all these files right here because they're going to be useful. Okay. So what I will do is like basically select all of this right here.
Okay.
So it selects everything and then go to this tool right here with the settings tool. more, and then you can basically do export. And then this is basically like name it how you want and then start to download it. And remember to put them in the right folder, like on your computer.
Thank you very much.
Yeah, of course, no problem. Does anyone has any question?
Thanks, Sarah. And just to confirm, will you share your slide deck with the community later today, just in case?
Yeah, sure.
Back
Yeah,
that?
I will.
Excellent. So I think Sarah will do that, and maybe Daniel, if you're still here, you can do that as well, or we'll just ask you. Any more questions for Sarah? Okay, so we are making good time here today. We have another break scheduled. We're running a little bit early, so let me ask Daniel and Daniela, are you okay if we take a quick 20-minute break here and then resume at 9:30 Eastern time?
Yeah, that's okay.
Okay.
Fine by me.
Excellent. So why don't we all take 20 minutes now and come back in 20 minutes for Daniel and Daniela's concluding lecture for today. See you all soon.
Thank you very much. See you soon.
Okay, everyone, welcome back. I hope I have Daniela and Daniel with me.
Hey.
All right, can hear you. And Daniela, how are you?
Good. How's it going?
All right, good. I can hear both of you. So Daniela and Daniel will be giving our last lecture of the day on single cell data exploration and quality control. So I will pass it on to the two of them. Thank you.
I think we're actually still just waiting for Daniel. I don't see him here just yet.
Apologies,
there
it
he
was the
is.
wrong Daniel.
Oh,
Apologies.
sorry.
It's okay. He's here now.
Yeah, I see your colleague. So good. All right, I think we're all set to begin then.
Okay. Hi again, everyone. So we're going to be doing the QC lab. Let me open the okay. So we're going to, I think, break out into two and work on this lab. I'm just going to give you an overview of what we're going to be doing, and then we can go to the breakout rooms and then come back together a little bit later. So first thing you want to do is go to File, Open File, and...
Hey, Daniel, we can actually see your screen.
Oh, let me try again. Can you see it now?
Yes.
Yeah. Thanks. Yeah, so yeah, you want to do File, Open File, and go to the HCA COVID-19 QC lab. So it's going to be this R markdown file. And basically we're going to be loading in the count matrix that we discussed. It's a data set from COVID-19, a COVID-19 study. We're going to be doing QC filtering and normalization on the data. we'll see different features of Seurat functionalities. We'll also try to understand better what the count matrix is, about different metadata and how we visualize them. So yeah, we're just going to go through all of that. And the first thing you want to do is change this home.dir to your home directory. So you can go to terminal, do pwd, and you can see the home directory that you want to use. So for me, it's home2/dchafamo, and I'll add that here. So make sure you change that to your home directory. And yeah, so we're going to execute these code chunks by running this play button. So here I'm going to do like play this code chunk, and I'm going to keep doing that for all the steps. We'll read these and run the code. Every once in a while, you'll see a to do. So that this was the first one, to change your home directory. The others are more about writing code. It's usually very straightforward, and you mostly repeat what you had in the previous code chunks. So we show you how it's done for one variable and ask you to do it for another variable. So it's mostly straightforward, but keep an eye out for the to-dos and try to fill those out. Yeah, I think that should be enough preliminary information. We can now go to our breakout rooms, and we'll help you get it going. And any questions that you have, you can ask any of the instructors, and I'll be in one of the breakout rooms. And then we can come back maybe after 30 minutes to go through it all together, and I can explain the outputs in more detail. Christine, can you help us go into break?
Excellent. Yes. So my colleague Tracy, are you there? Okay, I'm just going to go ahead and to assign automatically. All right, so what's going to happen is I'm automatically assigning folks to two rooms. They will open once I click the button. If you have any issues, Tracy and Elliot and I will remain here. So just come back here if you have any issues whatsoever. So Elliot, maybe you can test this for me. Are you able to let me know if the rooms are open, and is it working on your end? Okay. And I think you probably got a notification saying that you're assigned to a room, right? And you had to join. Okay, so you should all now see a notification to join the room that you've been assigned to. So please move out of this room and go to the room that I've assigned you to, please. Should all now be in breakout session room, so please go there. Okay, so for those still here, please join and go to the breakout session rooms. You should not be here now. Please join the breakout room. Irene, Amoaka. So there's still a few of you in this room. Please join the breakout session rooms that you've been assigned. Okay, Elliot, I don't know if there's anything else I can do for folks that are still in here, do you? Okay. All right, so for those of you still remaining in this room, you should, in order to carry out the hands-on activity, you need to move to your breakout session room. So please press join and go to the breakout session room. Okay, it looks like the rooms have now been closed. So we're back in the main hall. Daniel and Daniela, did you have closing remarks for today, or are we ready to release folks?
I think we wanted to go through the script just one last time before.
Absolutely.
Yeah.
Great. I'll hand it over to you, Daniel.
Thanks, Kristen. Sharing my screen again. Let me know if you can see it.
Yes, I can see it, Daniel.
Thank you. Hi again, everyone. I hope you are able to go through some of the script in the breakout rooms. I'm just going to give you a quick high level rundown of what we did in the script, and in case any people are not able to go through it. So the first thing that we do here, again, for people not familiar with our markdown, these gray patches are where the actual code, the R code is. So you have these three quotation marks, and then the R in curly braces, that marks the start of the code, and this marks the end of the code. Everything outside is a comment or a title just plain text. So we're going to be running the scripts using this play button here that runs this current chunk. Okay, so the first chunk we have here is to load the libraries. So this function just makes the methods in the libraries available to us. So we'll be using dplyr, Surat, and Patchwork. So to do our QC. So we're loading those libraries and we can execute that using this play button. So you can, in your own time, log in and keep running these run chunks, or you can come here into run and run all chunks below, and that should generate all the figures and create all the outputs so that you can just read through the script and look at the output. So you can just execute all of them and see the results. So we start again by reading our counts matrix. This is the table that I showed you in the presentation. We have cells here, and then genes on the rows. So cell AGAGT ... has 0 A1BG genes. So that's how you'd read this count matrix. And then we have our metadata, which is all the extra information that we have about our the samples from which each cell came from. So for each cell, we have the donor ID from which it came from, its species, sex, disease status, whether it's COVID positive, whether it's COVID negative, the age of the patients from which it was taken, the organ from which it was taken, and so on. So each cell will have this information in the metadata table. So we use those two to create our Surat object. So this function create Surat object will generate the Surat object in which we'll store our counts matrix, our metadata, and every other downstream analysis that we'll do. Again, we'll store it in Surat, in our Surat object. So you can read again more details about it using this, the help information to create a Surat object. So we are not filtering. So this mean.cell, min.features do some preliminary filtering. We want to do that ourselves. So we're setting them to 0 for now. So this creates a Surat object for us, and we can see that we have an object of class Surat in this Surat raw object. So the first thing we want to do is we want to try to understand our data set, see the number of cells and genes that we have. And to do that, we use this dim function, which gives us the dimensions, the number of rows and the number of columns of our Surat object. And we can see that we have 6,000 cells and 32,000 genes in this table. So that's our Surat object. We can see the count matrix again by using this get assay data function, which for the Surat object will give us the count table that we talked about before again. So we'll have the cells and the genes in the rows. So this is our a snapshot of our count matrix. And then the metadata, in order to access the metadata from the Surat object, you just do @ and then meta.data. So this syntax, your Surat object @ meta.data will give you the metadata table, and you can view the column names using column names. And so this shows you what metadata is available for you. So we're interested, for instance, in checking how many cells come from COVID positive samples and how many cells come from positive COVID negative samples. And in order to do that, we can use the table function. So table will count up the unique values for each column. So this column here, SARS-CoV-2 PCR status, tells us the PCR status of the sample from which a cell came from. And we can see that 3,500 cells came from COVID negative samples and 2,600 came from COVID positive samples. We have a to-do here to get the same kinds of values for the different sex values. So for male and female, how many do we have? All you need to do is copy this and change it from SARS-CoV-2 PCR status to sex or to any other metadata value that we have, and that will give you the distribution across the different categories. Okay, and we go to end-count RNA and end-feature RNA, which were calculated by Surat automatically for us when we created the Surat object. So in order to access a metadata column, we can use this dollar symbol. So when I do Surat raw end-count RNA, I'll get all the end-count RNAs for each participant. So this for each cell, sorry. This cell, for instance, has 5,745 number of RNAs total. So we can do different functions. We can apply different functions to this count. So min, max, mean, anything that we want to understand about it, we can use this column. And finally, we can use Surat's violin plot function VLN plot to generate a violin plot that we saw in the presentation. I find it helpful to focus on just the red distribution plot. So the way you'd read this is you can see that the red is like the density. So we have a lot of cells with around 3,000 to 10,000 number of RNAs total, and very few with values greater than 30,000. You can see it's very small thin here. Same around here. After less than 1,000, it's very much thinner, the red distribution marker. So we can see most of our data is in this region from 1,000 to 10,000. We can visualize the same thing for end-feature RNA. All you'd have to do is just replace this in the features argument. Instead of end-count RNA, you'd put in end-feature RNA. So violin plot takes the Surat object and a set of features. So here we did end-count RNA, you can do end-feature RNA. And we set log equals true here. So this is what generated this plot, but we can, by default, it is set to false. That just shows us this y-axis in normal without logging it. It's harder to see here because most of our data is located in this 1,000 to 10,000 region. But yeah, for the higher values, it's helpful to visualize without setting log to true. But for the lower values, it's hard to see here what the lowest values are. So here you can do log equals true. And you can see more clearly what the lower values, the distribution for the lower values are. Okay. And then finally, to generate our percent mitochondrial genes, we use this percentage feature set function again from Surat, which calculates the percentage of counts that come from genes matching this pattern. So we give it this empty pattern. All mitochondrial genes start with the empty prefix. So this will calculate the percentage of mitochondrial genes for us. And again, you can go ahead and plug in %.empty into these. You can copy and paste these and replace. So we do something like So we would come here and paste, and instead of, so run this first, and instead of ncount RNA, we can do %.mt, remove the log, and execute that. And now we have a % mitochondria plot. So very straightforward. %mt is now added to our metadata using this syntax, and so we can visualize it here. So another thing we can do is we can visualize multiple columns from our metadata table in the same violin plot. Just add a comma and add the different columns that you want to visualize. Here we do ncount RNA, N-feature RNA, and % mitochondria. We can also visualize again the joint plot of two features here, % mitochondria and ncount RNA, using feature scatter. So here you give it two features, feature 1 and feature 2, and it will plot those values against each other. So the way you would read this plot is you have this, every dot is a cell again. And for this dot, for instance, where my cursor is, we can see that it has 25,000-ish ncount RNA and around 18 or so % mitochondria. So we can see the value of both the % mitochondria and ncount RNA for every cell. And then the other cool thing we can do with violin plot is visualize the features that we've been discussing again, but grouped by different classifying metadata values. So here we use core cell annotations so that we can visualize the number of genes, number of UMIs, and % mitochondria for each cell type. So here group.by equals the core cell annotations. This generates for us the QC metrics for each cell type. So for B cells, this is the distribution of ncount RNA, same for basal cells, and so on. And again, repeated for N-feature RNA. And this is what you want to look at after you do your cell type annotation. You want to go back to your QCs and look at them again. Okay, and again, we can do this for donor ID. All you do is, again, copy this. And then change core cell annotation to just donor ID, and that will plot for you the QC metrics, but separated by donor ID. So for each donor, we have different the distributions for each QC metric. Okay, and then finally we do the actual subsetting here by using the subset method. So we tell it to subset by these cutoff thresholds. So N-feature RNA greater than 200 and ncount RNA greater than 400, and so on. So what this does is it takes our sort raw, and for each cell, it checks if this condition is true. So if any of this is false, it will remove that cell from our data. So if a cell's number of UMIs is less than 400, for instance, that cell will be removed. And if it's % mitochondria is greater than 40, it will be removed, and so on. So what we'll have in this raw object at the end of the day is our QC filter data. So once you've done this, you want to visualize how many cells you have, and it's also a good idea to, again, produce these figures and see after filtering, what does my data look like? And again, you can just run this. So I can copy this, first run the subsetting, and then paste my data, and then visualize this, recreate these plots. And now you can see that the highest % mitochondria is 40. If I set it to, for instance, 20, and run this again, and execute this plot again. Now we can see that the maximum percent mitochondria that we have in our data is 20. So everything that had percent mitochondria above 40 has been removed. And you can see same for N-feature RNA, which is 6,000, and counter RNA. The lower threshold that we set is 200 and 400. Okay, and the last step is normalization. We discussed what log normalization is. This is Surat provides us a method called normalized data, in which we can do CPM normalization and then log transform our data. So we give it the scale factor, which is the value by which we multiply after dividing by our by the total count for each cell. So it doesn't really matter too much, but 10,000, 100,000 would be standard values. And once we run normalized data, the normalized values will be stored in the data slot. So Surat raw, our assay, and then the data slot will give us our normalized values. And this is what we'll be using tomorrow for clustering and so on, all the downstream analysis that we'll be doing. If you have any questions or anything you want clarified, please let me know. Otherwise, we can stop here.
Great. So any questions for Daniel? All right. So I believe that this concludes day 2 of the workshop. So tomorrow we will begin same time as today, so 7 a.m. Eastern time, 12 p.m. GMT. Tomorrow will be the last day. And Or Aschenberg and Sergio will be presenting, and we'll also be sharing some work from the breakout sessions via group presentations. So please do show up tomorrow ready to learn and get to work. And if there's any questions or housekeeping that I'm forgetting, feel free to jump in. But if not, we will see you all tomorrow for our final day. Or anything that you wanted to say?
Nope. See y'all tomorrow.
Excellent. So thank you all and see you tomorrow.